Children, I hope you have gone through my previous video, and today we will continue further. Children, in continuing with the chapter Biomolecules, we have already studied about carbohydrates, proteins and enzymes. Now next we have to do nucleic acids, because hormones and vitamins, they have been removed from your syllabus in the reduced syllabus which has been sent by the council. So nucleic acids, the nucleus of the cell, that transmits inherent characters of individual from one generation to the other generation. This is called as heredity. Now nucleus consists of chromosomes which are made up of proteins and nucleic acids. There are two types of nucleic acids: deoxyribonucleic acid, which is called DNA, and ribonucleic acid, which is called as RNA. The repeating unit of nucleic acids, they are called as nucleotides. They are monomeric units and they are called as nucleotides. Therefore, they are regarded as polynucleotides because they are getting repeated. Now, a single nucleotide consists of a nitrogenous base, pentose sugar, and a phosphate group. Now, let us take them up one at a time. First, we start with nitrogenous base. Now, nitrogenous bases, there are two types of nitrogenous bases: purines and pyrimidines. Now, two purines are found in nucleic acids. They are adenine and guanine. Adenine is represented by A and guanine by G. Now, the second type of nitrogenous base is pyrimidine. Pyrimidine, there are three types of pyrimidines in nucleic acids. First is uracil, which is represented by U, thymine T, and cytosine C. Now DNA contains A, G, that is adenine, guanine, thymine and cytosine. So A, G, T and C. Whereas RNA contains adenine, guanine, uracil and cytosine. A, G, U and C. Alright, the chemical structure detail you don't have to go into because only the superficial information is there for your syllabus. Now next you have a pentose sugar. Second part of a nucleotide is pentose sugar. So there are basically two types of sugars present in nucleic acid. RNA contains beta D ribose, but in DNA there is beta D 2-deoxyribose. This is beta 2-deoxyribose and this is beta ribose. So ribose sugar is found in RNA and deoxyribose is found in DNA. The third part of nucleotide is phosphate group. So this phosphate group is responsible for linkage of a nucleic acid polymers, and it is bonded to hydroxyl group of sugars. So PO4, this is the phosphate group. Now a nucleoside, there are two terms here, nucleoside and nucleotide. Till now you were studying about nucleotide, which contains nitrogenous base, pentose sugar and phosphate group. Now nucleoside, it has only two components, pentose sugar and nitrogenous base. Phosphate group is absent in nucleoside, but it is present in nucleotide. Now depending upon the type of sugar which is present, nucleosides are of two types: ribonucleosides and deoxyribonucleosides. Obviously, if the ribose sugar is there, it will be called ribonucleoside. If deoxyribose sugar is there, it will be called as deoxyribonucleoside. A nucleotide contains all the basic components of nucleic acid, that is phosphate, phosphoric acid group, pentose sugar, and a nitrogenous base. And when a large number of nucleotides are connected, we get a polynucleotide. So this is about the basic components of nucleic acids. Now we come to the structure of DNA. Now DNA, deoxyribonucleic acid. It has a double helical structure, and the structure was given by Watson and Crick in the year 1953. Now this structure you have to study, not the chemical structure. Now they proposed that DNA polymer, they form duplex structure consisting of two strands of polynucleotide chains which are coiled around each other in the form of double helix. The nucleotides of each strand of DNA are connected by phosphate ester bonds. Base pairs of two strands, they are linked together through hydrogen bonds. Purine base of one strand is always paired with pyrimidine base of other strand. Like see, if G is here, then it will pair only with C. A will pair with T. Okay. Only possible pairing in DNA are between G and C. So between G and C there is a triple bond. And A and T. Between A and T there is a double bond. So there are two hydrogen bonds between A and T and three hydrogen bonds between G and C in DNA. Two of the helix, they are complementary and they are not identical. Diameter of the double helix is 2 Newton meter. This diameter is, see here, 2 nm. Alright, and the double helical structure repeats at an interval of 3.4 nm. So after every 3.4 nm, the double helix repeats itself. So when it completes one turn, so hence you can say that one turn is of 3.4 nm, and one turn corresponds to 10 base pairs. There are 10 base pairs in one turn. When DNA strands separate on heating, it is called melting, but on cooling, when they hybridize, it is called as annealing. The term is annealing, ANNEALING. The temperature at which both strands are completely separated from one another, this is called as melting temperature. DNA acts as carrier of genetic information from parents to offspring, and it is involved in synthesis of RNA. Next we come to the second type of nucleic acid, which is ribonucleic acid. Now it is found as genetic material in some plants and animal viruses, while in other living organisms it occurs with DNA. Each ribonucleotide consists of pentose sugar, nitrogenous bases, but the nitrogenous base here are A, G, C and U. Instead of thymine, there is uracil in RNA and phosphate group. Another difference is that RNA is single-stranded, whereas DNA was double-stranded. Except for certain viruses, all RNA are single-stranded. Complementary base pairing can occur between, the single strand can coil around each other. So complementary base pairing can occur between adenine and uracil and guanine and thymine. Cytosine. Thymine is absent. So between adenine and uracil also there will be a double bond, and between guanine and cytosine you already know there is a triple bond. Okay. Then there is a term DNA fingerprinting. This is a technique. The scientists look for a number of short fragments of DNA, and the base sequence of DNA is unique for an individual, just like our fingerprints. So apart from criminal investigation, DNA fingerprinting, it finds application in determining the paternity of an individual. Who are your parents? You and your parents will have the same DNA fingerprint. Identification of corpses and racial groups. Now the functions of nucleic acid. DNA is the genetic material of cell, forms chemical basis of heredity. It helps in transmitting characteristic features and from parents to offspring, and it maintains the identity of a species. During mitosis cell division, DNA duplicates and similar copies are transferred to daughter cells. Proteins are synthesized in the cell through RNA molecules, which in turn are coded by DNA. So the process of synthesis of RNA from DNA, it is called as transcription, and process of formation of protein from RNA, it is called translation. So this depicts the role of nucleic acid in protein synthesis. So certain terms you need to remember, like annealing. Annealing is, when after melting the DNA hybridizes. So that is annealing. They again hybridize on cooling. So that is annealing. Then one more term is transcription. That is the process of synthesis of RNA from DNA, and translation, process of production of protein from RNA, that is translation. So few things which you need to remember, and that is all in this chapter. Go through the question answers. Restrict yourself to what has been taught, because lot of it has been removed from your syllabus. This finishes this chapter.